Hello students, today now we are going to discuss about genetic code and protein synthesis. So before going that one, you see generally the cell means it is a structurally functionally basic unit to all biological living organisms. Generally it is a two parts is there. The first part is the cell wall, second part is the protoplasms. So these protoplasm is contain different type of cell organelles like Golgi bodies, ribosomes, so many cell organelles is there in that one. And in spite of center part is the most important. The center part we call that is a nucleus. So generally this nucleus, here you are looking the four parts. So one, it is a nucleus envelope. So where we are looking in the prokaryote, so where we are looking the eukaryotic, it is the four parts is there. So one, it is a nucleus envelope. Second, that is the second one, that is a nucleolus. Third one, that is in chromatin network. So after the chromatin network we are looking the DNA. So what is the meaning of in DNA? DNA means that is of deoxyribonucleic acids. So generally when we are looking in the DNA, so one, it is in a proteins, second that is of in sugars, so third one that is in nitrogen bases. So that is in nucleotide is there, so nucleosides is there. So these DNA is contain the proteins. Above the DNA we are looking in the proteins. So now you see we are going the detail explain our senior professor, so Mr. Padala Tirupati sir, he will give all the details about. So what is the genetic code? Code means above the DNA we are looking some little type of the characters. So he will give the explains. Sir, what is the meaning of a genetic code, and what is the, when we are looking in the above the DNA proteins, how the proteins are synthesized? So please explain that one, sir.
Thank you, Ramakrishna sir. Genetic code is the blueprint of life. So genetic code, it contains the different nucleotides. The nucleotides are three nucleotides are code 1 amino acid. It contains A, U, G, C in DNA, and RNA also contains nucleic acids, nucleotides, that is A, U, G, C. So 64 codons, it codes 21 amino acids, and the 3 codons does not code any amino acid. That codons we can call as stop codons. Also we can call as nonsense codons. The best examples are the first codon, that is codes methionine, it is AUG, and the 3 codons does not code any amino acid, that is UAA, UAG, UGA. These codes we can call as stop codons or termination codons or nonsense codons. Including that codons, some of the codons like UUU and UUC, these codons codes for amino acid, that is phenylalanine. The features of the genetic code are, it is a triplet codon. Triplet codon means each amino acid coded by a sequence of 3 nucleotides. So that's why we can call as triplet codon. Our genetic code is degenerate codon or degeneracy codon, which means multiple codons code for one single amino acid. Our genetic code is universal. That means any type of organism, including prokaryotes, eukaryotes, higher organisms, from lower to higher organisms, our genetic code is accepted for universal. Our genetic code is non-overlapping codes. That means read sequentially without overlap, and also our genetic code is unambiguous. Each codon codes for only one amino acid. It contains start codons and stop codons. Start codon we know that is AUG, methionine, and stop codons are 3 codons, that is UAA, UGA and UAG. Here we see that the codons UUU codes for phenylalanine and AUG codes for methionine, UGA codes for stop codon. Then afterwards the our genetic codon codes for 61 amino acids. I am showing these different types of codons codes for different amino acids. In this AUG codes for methionine, it is the first codon, and the last and stop codons UAA, UAG, UGA. This codon does not codes for any amino acid. This is most important. And what is the application of this genetic code? We know that today.
Sir, excuse me, sir. You see here generally we are looking the DNA and RNA.
Yes.
So what is the difference between DNA RNA means, sir?
actually each organism contains two nucleic acids.
Yes.
That is deoxyribose nucleic acid and ribose nucleic acids.
Okay.
DNA is acted as a genetic material majority of the all organism, but RNA is also acted as some of the viruses. It is acted as a genetic material. Recently we know that pandemic situation due to the coronavirus contains RNA is a genetic material. So some of the microorganisms, some of the lower organism contains RNA as acted as genetic material, but most of the organism contains DNA.
So generally DNA, RNA mean DNA is genetic material, RNA is not a genetic material. So where there is absence of in DNA, there we are looking in, so RNA acting as a genetic material.
Yes,
sir?
yes.
Yes, sir, very good, sir.
Including the applications of the genetic code, it is most important for gene therapy. Nowadays we are facing so many genetic disorders. These type of disorders can modify, or even though the treat by this gene therapy due to the involvement of this genetic code. And also the most important genome editing technology, that is CRISPR-Cas technology, and also DNA fingerprinting technology, synthetic biology. These are the most important applications of the genetic code. Some of the key features of the genetic codes we have already discussed, that is triplet codon and universal codon, and contains stop codons and even though the stop codons. The applications and significations of the genetic code is most of the any organism from prokaryote to eukaryote, protein synthesis is most important metabolic phenomena, and also the genetic process including DNA, the genetic material from DNA to transfer to RNA and RNA to protein. So that we can call as the protein synthesis. It is also controlled by the genetic code, and some of the mutations and diseases also occurring due to the some of the problems raised in the sequence of the genetic code. So that's why we are already observed in the chromosomal mutations or genetic mutations, even though some of the genetic disorders we are observed due to this the code sequence is lost. And today we are observed so many genetic engineering and gene editing technologies in the process of biotechnological applications, including synthetic biology and medicine. The entire genetic code is very important phenomena.
So here generally protein synthesis. Protein synthesis means DNA to RNA, RNA to next it goes to the proteins. We call that as protein synthesis.
Yes.
Sir, here mutations. So what is the meaning of mutations? How can we recognize in the diseases, sir?
Yes, the mutations, it is a sudden changes occurring in the sequence of genetic material that leads to the problems in chromosome parts, or even though the DNA or genes, that we can call as mutations. So mutations is identified by using of these genetic code applications, whether the gene code or sequence of the gene whether lost or even repair, but if even though mutations has removed by the applications of these biotechnological applications like CRISPR-Cas technology, that is nothing but genome editing technology.
Okay.
So in this regard, definitely we will going to discuss about the most important biological process, that is protein synthesis. So without proteins our body does not build. So our body is built with the proteins. So protein synthesis is the biochemical process involvement of the nucleotides from genetic code. Here protein synthesis has the essential for cellular structure, function and regulation. This process occurs all living organisms, prokaryotes and eukaryotes. Major two steps are involving in this protein synthesis, that is transcription and translation.
Sir, here without proteins there is no DNA. Proteinia is the most important. Yes, sir?
Yes.
Yes. Generally protein synthesis, it is a two types is there. So one is a transcription, second that is of in translations.
Yeah.
Sir, please.
So
sir.
these two steps are most important for the protein synthesis. Along with these two synthesis, additionally several post-translation process modifications is also most important for the understanding of the complete synthesis of the proteins. Okay. Next, then in these stages of protein synthesis, first step, that is we can call as transcription. any type of organism, all eukaryotic organism, transcription takes place in the place of nucleus, but in prokaryotes there is no definite nucleus. That's why transcription takes place in the place of cytoplasm. This is most important point, my dear friends. Remember here transcription, where transcription has occurred. Transcription has occurred in eukaryotes in nucleus, whereas in prokaryotes cytoplasm. This is a major difference between pro and eukaryotes. In this transcription process, majorly 3 steps we are observed in the transcription, that is initiation, elongation, termination. Initiation is the first step in the transcription. In this transcription, the enzyme, that is RNA polymerase, binds to a specific DNA sequence called the promoter region. So it is the first and foremost reaction which we are observed in the process of transcription in the nucleus of the eukaryotes, but in cytoplasm of the prokaryotes. In this process, the DNA double helix unwind, or the coiling of the double helix DNA is going to unwind to expose the template strands. This is the most important step in the transcription, that is initiation. After initiation, so many enzymes are involved in the process of the elongation process. In this elongation, RNA polymerase reads the template strands of DNA synthesis and the complementary mRNA strand by adding nucleotides like adenine, guanine, thymine, cytosine of the DNA. But whereas in mRNA strand, it is template to the DNA. That means the mRNA contains, or mRNA nucleotides are adenine, uracil, cytosine and guanine. Thymine is absent in mRNA, but thymine is present in DNA. This is the difference between mRNA and RNA nitrogen bases. My dear students, remember, uracil is present in RNA, but thymine is present in DNA. In this process, elongation process has taken so much of time. Afterwards elongation, termination process has stopped. Then whenever the genetic code is already, the sequence of the nucleotides already decided, whether it is codes for the mRNA strand is enlarged or is going to stop. But whenever the termination process has started, the transcription continues until RNA polymerase reaches a termination signal. Whenever RNA polymerase reaches the termination signal, automatically the transcription process going to stop. That we can call as termination. The newly formed pre-mRNA is released from the DNA from nucleus of the eukaryote in the cytoplasm of the prokaryotes. After the transcription process, some of the important processes or important metabolic processes which we are observed in the after transcription, that we can call as post-transcriptional modification in eukaryotes. Especially eukaryotes, definitely we should observe, we should identify these 3 important biochemical processes has done or not. That is, number 1, 5' capping. It is very, very important in the process of after transcriptional process or post-transcriptional modification. In this 5' capping, a methylated guanine cap is added at the 5' end for the stability and ribosome binding. Then second important biochemical process in the molecular structure, that is polyadenylation. In this polyadenylation, the polyadenylation tail or poly-A tail, we can call as poly-A tail. This poly-A tail contains multiple adenine residues is adding to the 3' end. It is most important for the protein. And the third important post-translational transcriptional modification, that is splicing. Very, very important. Whenever the splicing has lost, the genetic disorder or mutation has occurred. So when the biochemical or the molecular splicing is applied, then automatically if any changes has occurred in the genome sequences, then automatically it contains coding sequences and non-coding sequences. These coding sequences and non-coding sequences are most important, but in the process of splicing, the coding sequences are joined but non-coding sequences are removed in the process of splicing. In this splicing, what has occurred? Introns are nothing but non-coding regions. These are removed in the process of splicing. But whereas the exons, these are the most important nucleotides or genome sequences which has joined to the mRNA. So this is the most important, the molecular and biochemical process of the post-transcriptional modifications.
Okay.
Afterwards, the second major step or the second phase, that is we can call as translation. My dear students, remember, transcription completely observed or occurred in the place of nucleus in eukaryotes. Prokaryotes, cytoplasm only. Here, my dear friends, remember, translation. Translation means the genetic material or mRNA, the nucleotides of the mRNA is transferred to code to number of amino acids. It reaches the polypeptide chain. Finally, protein synthesis has occurred. This we can call as translation. In this translation, we are observed prokaryotic organism, translation and transcription, both the processes of the protein synthesis has occurred in the cytoplasm, but this translation definitely we can observe in the eukaryotes, ribosomes, at the same time cytoplasm. Here, remember my dear students, ribosomes are two types. Actually, Ramakrishna sir previously told that ribosomes are two types, that is 70S ribosomes and 80S type of ribosomes.
Yes,
70S type of ribosomes which we are observed in the prokaryotes, whereas 80S type of ribosomes which we are observed in eukaryotes. This is the main difference between pro and eukaryotic ribosomes. My dear friends, remember, ribosomes are the very primitive cell organelles in the entire living organism. Why? Because whenever the pre-life form or primitive earth, the ribosomes are present. Whenever the organism, lowest organism, that is blue-green algae member, it also contains 70S type of ribosome. But whereas in eukaryotes, 80S type of ribosomes are present. These ribosomes are most important for the translation process, including, remember, the translation has also contains 3 steps like transcription. It is also contain 3 steps. Number 1, initiation. Number 2, elongation. Number 3, termination. Same, but the major reactions or major enzymes are involved, different types of enzymes are involved in the translation process in eukaryotic organism. Here, the initiation of the translation, number 1, the small ribosomal subunit. Remember, 70S ribosomes contain 30S and the 50S type 50S subunits. Ribosome contain 2 subunits. First one, smaller subunit is 30S and larger subunit is 50S in prokaryotic organism. But eukaryotic organism ribosomes contains 2 subunits. The smaller subunit is 40S, then larger subunit is 60S. So these 2 subunits are most important in the process of initiation of translation. In this translation, ribosomal subunits binds to the mRNA at the start codon. Any type of organism, whether eukaryote or prokaryote, start codon is AUG, that codes methionine amino acid. But in prokaryotic, AUG codes for methionine in eukaryote, but GUG codes for methionine for prokaryote. This is a smaller difference between pro and eukaryote. In this, a transfer of RNA, that is tRNA carrying methionine, that we can code for MET. Methionine means MET, binds to the start codon, that is the initiation codon. The large subunit, smaller subunit binds with mRNA, first codon. The large subunit that attaches to the forming a functional ribosome. It is the initiation process of the translation. But the second step of the translation, that is elongation. The ribosome moves along the mRNA reading codon. Reading codon, remember our genetic code is triplet codon. BRAOU has coded, our genetic code is triplet codon. So the triplets of nucleotides reading codons, it is in the process of elongation. Each codon is recognized by a tRNA carrying a specific amino acid. So tRNA is most important involved in the carrying of specific amino acid. Then the ribosome catalyzes the formation of peptide bonds between amino acids. tRNA means transfer RNA. This transfer RNA move through the ribosomal sites. The ribosomal sites are A, P, E. A means amino acyl sites, and P is peptidyl site, and E is, that means exit. The first one, amino acyl site, tRNA carrying the next amino acid arrives from the mRNA. Then P, peptidyl site, it is a growing peptide chain. So one amino acid is going to attach then another amino acid, that is the site, the site of the ribosome, that is peptidyl site, it is a growing polypeptide chain is held. Then exit site, it is empty RNA, empty tRNA, that means it is exists from the ribosome. The last step of the translation, that is termination. In this termination process, when the ribosome reaches the stop codon, actually we are observed in the mRNA, it contains different nucleotides. These nucleotides codes for different amino acids. Here, whenever the ribosome slowly moves towards the growing from one place to another place, it moves towards the progressive manner. Whenever it reaches mRNA, that is ribosome on the mRNA strand, it reaches the stop codon, that is UAA, UAG, UGA, then automatically translation is stops. Remember, my dear friends, when the ribosome reaches, if ribosome not reaches the UAA, UAG, UGA, then automatically translation process continuously it is growing. So whenever we reaches or whenever we fulfilled, whenever we needed for the stopping the translation process, at the time UAA, UAG, UGA, these are the stop codons has occurred in the strands of mRNA, then automatically translation process stops. And a release factor binds at the time. Remember, whenever RNA or ribosome reaches the stop codons, at the time release factor binds, then causing the ribosome to disassemble and release the newly synthesized protein. So one protein is synthesized in ribosome. Ribosomes also we can call it as protein factories. So whenever one protein is synthesized, this entire process, molecular biology, this transcription and translation continuously without any interruption, it continuously growing on or continuously reaches, then finally protein has synthesized. So this is the most complexity process of the protein synthesis. Translation is the 3 steps, completely very complex process of the protein synthesis, my dear students.
Sir, where the stop codons, that is of UAA, UAG and UGA.
Yes.
So when the codons is come there, that is stopped of translations.
Yes.
Means translation is completed in that one.
Yes.
Yes, sir. This is of translations.
Why? Because
Yes.
these UAA, UAG, UGA does not code any other amino acid, any amino acid.
Any other amino acids.
That's why these UAA, UAG, UGA called as nonsense codons or stop codons or termination codons.
Okay.
It terminates the protein synthesis.
Okay, sir. Yes.
Okay. Then after translation, most important translation modification, post-translation modifications also we are observed in the protein synthesis. That is number 1, folding. Folding is assisted by chaperone proteins. After translation, after translation, the post-translation folding is assisted by chaperone proteins. Chaperone proteins are most important proteins which is useful in the involvement of the post-translation modifications. Then cleavage. Some of the proteins are cut into functional fragments. Functional fragments are most important for the other biochemical process of the all living organism. Glycosylation. Glycosylation, it is addition of carbohydrate groups. We know that carbohydrates, they are simple carbohydrates or complex carbohydrates, we know that. But the glycosylation process, it is addition to the carbohydrate groups. Yes. Then phosphorylation, it addition to the phosphate groups. Then lipidization, addition to the lipid molecules. Okay. Then yes. What are the key molecules involved in protein synthesis? DNA, RNA, tRNA, yes. ribosomes, enzymes. These are all involved in the protein synthesis.
So, sir, these are all the most important key points of when the protein synthesis. Yes, sir?
Yes, yes,
sir. So, that is of in mRNA and tRNA, rRNA and ribosomes, enzymes.
Yes.
So, these are all most important involved to the when the protein is going to synthesis.
Yes,
yes sir?
Yes.
Yes, sir. Now, next, what is the differentiation between?
The major differences between pro and eukaryotic protein synthesis.
Okay.
The main features of this pro and eukaryotic protein synthesis, number one, we are already discussed location. Location of the prokaryote is always, whether the transcription or translation, is itself cytoplasm. Remember, my dear students. But location of the protein synthesis in eukaryotes, nucleus, transcription observed in the nucleus, at the same time translation observed in the cytoplasm. Then mRNA processing. mRNA processing in prokaryotes, there is no modifications observed in the prokaryotes, but eukaryotes, it is a very complex process, that is capping, polyadenylation and splicing. Ribosomes involved in prokaryotes, 70S type of ribosomes, but eukaryotes, 80S type of ribosomes. Transcription, translation simultaneously, continuously in the prokaryotes, but eukaryotes, separate process. Nucleus site is different from the cytoplasm. So that's why transcription and translation both are separate process. Then initiation factors. Initiation factors are very little amount of the initiation factors are necessary for the prokaryotes, but more number of the initiation factors are necessary for the eukaryotic protein synthesis. And what is the regulation of the protein synthesis? Remember, my dear friends, the transcriptional control and mRNA processing control, translation control and post-translation control, these are all involved in the different levels of the protein synthesis. Finally, we can conclude that protein synthesis is a fundamental process ensuring that genetic information is expressed as functional proteins. So, it is nothing but functional proteins are nothing but it is a hidden genetic makeup, hidden genetic material. It is involving in the control of the entire protein synthesis. Understanding that protein synthesis is a crucial in the fields like genetics, biotechnology and medicine. Any type of biochemical processes is controlled by the genetic code and protein synthesis.
Thank you, sir. Nice explanations. What is genetic code and what is protein synthesis, and in spite of when the protein synthesis is using to, so very much that is of biotechnology and medicines. Thank you, Tirupati sir. Nice explanation of protein synthesis, sir.
Thank you, sir.